Alrighty, hello all. Welcome to our BISC202 genetics final review session. We're just going to go straight through the practice exam that I emailed to you, and also that is on Canvas. There is a key on Canvas, and I highly encourage you to look that over, but please take the practice exam before watching this video so that we can go through each question together. So I'm going to go through all the questions, and then I'm going to go through PEQ 13, which I had some requests for. That's the game plan. Okay, so question 1, name the enzyme that synthesizes RNA during DNA replication. So when I'm reading this question, I'm going to highlight DNA replication. That's our context, and I'm going to bring to memory this image. So when we're looking at DNA replication, we have this image, and remember that the first step for DNA replication requires an RNA primer for DNA polymerase. So this question is really, where does RNA play a role in the context of DNA replication? And that's in the primer situation. The primers are laid down by primase right here. Primase. So that's the correct answer to this question. Question 2. Which of the following are needed for termination of translation? Circle all the correct answers. So this is the important part, termination of translation. This is the image that this termination of translation process brings to mind. Remember that release factor? It acts like a tRNA, but it's not. It interacts with a stop codon, but it doesn't have any amino acids attached to it. So, to stop translation, the ribosome needs to encounter a stop codon. And a protein called a release factor, this guy, recognizes the stop codon, and that's how the end is signaled. So we're going to skim through this. The termination sequence, that's a transcription thing. Termination tRNA is inaccurate. It's a release factor. It's not a tRNA. It's actually a protein that isn't made out of RNA. And clearly, because it's not tRNA, tRNA synthesis is not part of that process. So the correct answer is B and D, stop codon and release factor. Question 3. Focusing on the interactions between RNA molecules during transcription, where does a new RNA nucleotide attach to a growing RNA strand? And it specifically uses the word exactly here. So these are the things that I highlighted. We're talking about RNA transcription, so the making of RNA based on DNA, and we're talking about a new RNA nucleotide attaching to the growing strand. So this is the mnemonic that I suggest for this. There's read up and write down. So the RNA polymerase reads from 3' to 5' and builds or writes from 5' to 3'. So when you're adding things, you add things to the 3' end. And the 3' end is the OH group. The way that I remember that is that O and 3 are both round, and 5 and N are both pointy. But that's how I do it. You can figure out a different mnemonic. Synthesis always occurs in the 3' direction, and it's the OH group that is that 3' end of the RNAs that builds the peptide bond. And that's where a new phosphate group attaches. So this is the correct answer, and you need to have 2 aspects to get a full point. There's 3', which is an OH group. Cool. Question 4. How many peptide bonds can be formed during translation of a single RNA molecule if the cell does not have any EFG? So these are the important parts. Translation without EFG. And in order to figure this out, we have to remember what EFG does. So here's another image of the ribosome and translation and those active zones with tRNAs in them. We have our special beginner tRNA that doesn't need to advance. It actually starts in the P region, and then the ribosome kind of collects around it. And then a first tRNA comes in attached to something that is not that methionine of the starter tRNA. That first tRNA comes in, it is attached to amino acid. These two can connect, and that whole thing happens up to this point before it needs to advance. Before EFG is necessary to advance the tRNAs from PA into EP. So this is what the EFG does. It pushes it this way, and it advances the whole thing forward. And that all happens after the very first amino acid transfer from this guy to this guy. So, in short, the first peptide bond will occur, and the answer is B, 1. You'll only get one peptide bond happening. Question 5. The DNA double helix below is undergoing DNA replication. Circle a nucleotide on the original DNA strand that will not have a new nucleotide base paired with it once this DNA replication is finished. So, these are the important parts. We're in DNA replication, we're circling a nucleotide precisely. We're going to work on the original strand, which in this image is black. We're not doing any of the blue and red, because we're working with the original strand. And you're trying to find a part that won't get a new DNA associated with it. So we're not going to get a pairing there. So if you remember the telomere problem, the original DNA sequence on the 3' ends, they'll get primers. But those primers can't be replaced by DNA because there's no 3' end for that DNA to attach to. So there's not going to be any base pairing associated with that 3' ended primer, which is at the very start of the lagging strands. And because we're in this replication bubble, that's going to be these 3' ends here. So this is the correct answer. You were expected to circle these two things. Question 6. Which of the following structures help a prokaryotic cell ensure that transcription begins at the correct location? Circle all correct answers. So these are the important bits. We got prokaryotic, we've got transcription at the beginning of transcription, and we're asking about location as opposed to timeline. So, which structures are a prokaryotic transcription initiation process at the beginning? This is a sigma factor thing. It's what binds to the Tata box in the promoter region to recruit RNA polymerase for transcription. And the other three things are translation terms. So this is transcription, that's translation. The start codon is also a translation thing, that's important for translation. The initiator tRNA is translation, it's this sigma thing that's the other important part here. So the answers are A and E. Question 7. Which part of a gene controls when that gene gets transcribed in both prokaryotes and eukaryotes? When transcription, as opposed to location, now we're talking about timing. Okay, so this is what we looked at earlier. Same image. The gene can only be transcribed when the promoter is exposed. Other regulatory molecules can interact with that promoter to affect how much transcription occurs at specific times by determining how exposed the promoter is. So, the promoter is the important part. All the other things, that doesn't have anything to do with timing, that has nothing to do with timing, the Tata box is a location thing, the sigma factor is a location thing, we just talked about those. Neither of those are timing. The promoter is the only one that determines when a gene is transcribed. Okay, 8. If a tRNA molecule has the anticodon GUA, what amino acid should be attached to it? Okay, anticodon GUA. Anticodons are complementary to codons. and here's our codon table. We're looking for an amino acid that's associated with the complementary process to GUA, which is CAU. So if we look, starting at the first letter, it's C. Second letter is A. CAU is his, which is histidine. Question 9. What kinds of mutations did Brenner and Crick use to prove that RNA was transcribed 3 letters at a time, i.e. that each codon consisted of 3 letters? So, even if you've completely forgotten about how Brenner and Crick did their experiment, or even who Brenner and Crick are, we can still use contexts to figure out the answer to this question. Like, we're asking what types of mutations could be used to determine the nature of a codon. So, I am thinking, how do we mess up a codon? How do we affect change by changing a codon specifically? So these experiments did include single nucleotide insertions and deletions, but really any mutation that causes a frame shift would work in these experiments. So the answers are C and D, deletions and frame shifts. None of these 3 change codons specifically, change the frame. Just C and D do. So, question 10. If you randomly synthesize RNA strands in vitro using a 1:3 mixture of G and U nucleotides and then translated those strands into proteins with amino acids, what proportion of amino acids in the resulting polypeptide chains would be tryptophan? So, we've got a 1:3 mixture of G to U nucleotides that we're going to translate, and we want to know what proportion of the amino acids will be tryptophan. So let's quick skim looking for tryptophan. Aha, there it is, TRP tryptophan. There's only one codon, it's UGG. So, keeping that in mind, 1:3 means 1 G for every 3 U's. So, for each individual nucleotide, there is a 1/4 chance that you'll have a G and 3/4 chance you'll have a U for each individual nucleotide. So we're talking about a codon. We have to go slot by slot for the 3 codon slots. And again, we're remembering and versus or. And is multiply, or is add. So in order to have that UGG, this is an and scenario, because you need to have that first one being U, and that second one being G, and that third one being G. So the chance for the first one being U is 3/4, and the chance for the second one being G is 1/4, and the chance for the third being G is 1/4. And because all 3 need to be true, we're multiplying. So when you multiply these 3 together, you get 3/64. So that's how you do that. All right, true-false lightning round. Question 11 to 13. Viruses contain fewer genes than prokaryotic cells and eukaryotic cells. Correct. Viruses do contain fewer genes. They don't even have enough genes to replicate themselves. Next, the poly A tail is added to prokaryotic RNA transcripts to protect them from degradation by exonucleotides. True or false? It's false. And the key word here is prokaryotic. To make it true, you'd change prokaryotic to eukaryotic. And this is because, remember, the poly A tail in eukaryotic RNA transcripts does absolutely protect them from degradation by exonucleases, but that's not necessary in prokaryotic RNA transcripts because prokaryotic RNA doesn't go anywhere. It doesn't need to be protected by exonucleases because it gets translated as it's being transcribed in the same location. Next, a single base pair insertion always causes more changes to the amino acid sequence of a protein product than a missense mutation when these missense mutations occur within protein coding regions of a gene. This is true. And the important aspects are single base pair insertion versus missense mutation. A single base pair insertion causes a frame shift. A missense mutation is a point mutation where one amino acid changes to a different amino acid. That's the end result, but it doesn't change all of the amino acids, just the one in the final protein. Whereas a single base pair insertion will change all of the codons downstream because it's causing a frame shift. So that's true. All right, question 14. Describe two similarities between the mechanisms for DNA replication and bacterial conjugation. Okay, so similarities, mechanisms. So here is our conjugation image, and here is our replication image. And right off the bat, if we remember, both have consistent points of origin. So we've got a point of origin, an ORI-T, if you will, here, and in prokaryotic replication, you also have an origin of replication. Also, both separate double-stranded DNA into single-stranded DNA. Here we have our F factor separating out, and a single strand comes in and gets replicated. Here we're splitting the original DNA, and then a second is coming in underneath. So we're separating that double-stranded into single-stranded, and you need a mechanism for that. And as mentioned earlier, both are building new strands from that older strand, from a template, and both are going to be using DNA polymerase to do that. I'm sure there are others. These are just 3 that came to mind. So if you thought of others that don't fit this, go look them up on the internet, make sure that you're right. But you're probably right, because you're smart and I believe in you. 15. Population, oh yeah, this is a giant one, I forgot about this. Okay, so for this giant question, I'm going to first look at each chunk on its own. So we begin with the background context. We have a population of fruit flies that exhibit negative assortative mating based on eye color phenotypes, and these are the phenotypes. So this is the important part. Negative assortative mating. And question A is, what overall effect should this negative assortative mating behavior have on the frequency of the alleles in this population, assuming Hardy-Weinberg, and that he's asking you to justify it. Here are the important parts. We're looking at overall effect. We're still talking about negative assortative mating. We're talking about allele frequency, and we're assuming Hardy-Weinberg equilibrium, which means 2 things. Closed system, no active evolution, besides what we're saying. So there's negative assortative mating, nothing else is going on, no funny stuff. So in Hardy-Weinberg equilibrium, negative assortative mating specifically doesn't affect frequency. It does change the arrangement of the alleles within the different phenotypes. But because nothing is actually being selected for or against, and nobody's dying in the same way, so there's no pressure against one allele or another allele, we're not going to see any changes in allele frequency. So C, stay the same. Here's B. Same conditions. What overall effect should the negative assortative mating behavior have on genotypic frequencies of the population's eye color gene? Justify. Okay, so exactly what we're talking about, but instead of allele frequencies, we're talking about genotype frequencies. They still want to justify. So when we're talking about negative assortative mating, that means that the flies are attracted to different phenotypes of themselves, from themselves. So a red-eyed fly will be attracted to a yellow-eyed fly and more likely to mate with it. That means that the 2 different homozygotes are more likely to interbreed, which will result in overall greater proportion of heterozygotes. Heterozygotes will also mate. They're going to preferentially mate with homozygous recessives, and those offsprings will be half heterozygous and half homozygous. So this overall will lead to an increase in the frequency of heterozygotes. and a decrease in the frequency of homozygotes. And this is the most important part, this part that different homozygotes will interbreed, which will result in more heterozygotes, because E will preferentially mate with e, resulting in those heterozygotes. So that's why there's going to be an increase in those heterozygote frequencies and a decrease in the homozygous frequencies of both alleles. Okay, 16. Predict whether attenuation of the tryptophan operon will increase, decrease, or stay the same when the following changes are applied. Justify each of your predictions. First one, important parts. Predict whether attenuation of the tryptophan operon. So we're talking about attenuation, we're talking about tryptophan operon, we're talking about various conditions in that context. And you have to justify things. So it's not just the answer, you have to say why. So this context is increased concentration of glucose. Well, glucose is the carbon source for cells. Tryptophan operon makes an amino acid, tryptophan. There's no direct link between the need for carbon sources and the need for tryptophan. And as a result, there is no relationship between glucose levels and regulation of tryptophan attenuation. So the answer is going to be no effect on attenuation in this one. Next one, decreased concentrations of tryptophan to very low levels. Okay, so we've got decreasing tryptophan, and we know, you and I know, that there's a different response to high levels of tryptophan, low levels of tryptophan, and very low levels of tryptophan. At very low levels of tryptophan, we're going to see attenuation because the ribosome is going to stutter at that spot on the one slot here. It's going to stall because it will struggle to find available tryptophan attached to tRNA in this slot because there are those tryptophan codons. That's going to result in the anti-termination hairpin, which is not the one between 4 and 3, which is the termination hairpin. You need to know this. You need to be able to draw this, so make sure you can do that, because on the test, this image will not be there. So you're going to need to know that attenuation stops or slows the production of tryptophan. When tryptophan levels are low, the ribosome is going to get stuck on region 1, which will open up the hairpin loop that would normally cause attenuation, thus preventing attenuation from happening. Therefore, this will result in a decrease in attenuation and thus an increase in tryptophan synthesis. So go back, make sure you understand those details because that's going to be an important aspect of this test. Okay, 17, missing gaps. Fill in the missing gaps. This is the blood type thing. If you remember, this was a codominant phenotype. And we have the phenotypic frequencies of AB blood type and O blood type, which, if you remember, O is 2 little I alleles. These are the alleles we're working with. There's 3 of them. The frequency of each allele, what we have is the IA allele has a 0.2 frequency. So the first step is this one. We're going to assign IA to little p, IB to Q, and lowercase I, our recessive allele, to R. And step 1 is to find R. We know that O is our phenotype, which is R squared. Therefore, R is the square root of 0.25, which is 0.5. So that's R. And we know what IA is, which is P. So P plus Q plus R equals 1. We know P, we know R. We start with 1, we subtract P and R, and we get 0.3. So I'm going to take this 0.5 and these 0.3, and I'm going to put them where they belong. So R, 0.5, is going to go here, and Q is going to go there. See that? See that magic that happened? Hope you're following. Next, we're going to build a Punnett square. This is how I would do it. I'm also going to show you the mechanism for using the Harvey-Weinberg equation. But let's first just talk about the Punnett square, which is filling in the gaps. And in the Punnett square, again, we're going to use these and as multiply or as add situations. And we're going to start by doing the and, which is multiplying, by taking these allele frequencies that we have. So if you note, this is the allele frequency. See that? See what we did there? We carried it straight down. This is the same thing. And in order for this genotype here to exist, we need an IA and another IA. It's an and, so we're multiplying. So this is going to be 0.2 times 0.2, which is 0.04. Same goes for all of the other squares. We're going to multiply them together to find what's going to be in them. And this is what that looks like. And I'm color coding it to show you where I put things, because this is not genotype frequencies, these are phenotype frequencies. So in order to understand what the chances are of an individual having a blood type, you have to take all of the different A genotypes and add them together, because you could have an A blood type with this genotype, or you could have an A blood type with this genotype, or you could have an A blood type with this genotype. So you have to add them together to get the total frequency of getting an A blood type. And the same with B. You could have this B blood type, or this B blood type, or this B blood type. They're all B. You have to add them together. And you can check your work by looking at the slot that you already know, the AB blood type. There's 2 of them, and 0.3 times 0.2 is 0.06. And so that these two, if you add them together, you get 0.12. So it works. Let's talk about the Hardy-Weinberg equation. This is the one that I'm bad at because math is terrible and I hate it. To get blood type A, phenotype, you take the 2 things that would result in blood type A, the 2 phenotypes that would. So P squared would result in blood type A, and 2PR would result in blood type A. This is that heterozygote with P and R. There's 2 options, so it's 2PR. So P is 0.2. If you square it, you get 0.04. And 2PR is that. And together, you get 0.24. You could do the same thing with B, but this time using Q instead of P for that B blood type phenotype. Alternatively, all of these different blood types add up to 1, so if you've got everything but B, you can add them up and then figure out what's left. What's left is B. This is B. My cat stole a chicken bone. I'm gonna go rescue it. Cat! You're not supposed to be eating that! I'm back. Question 18. Merodiploids. Remember those? Ugh, those. Okay. When a merodiploid with this genotype is in an environment with high concentrations of lactose and glucose, how much expression of the Z structural gene would you expect to observe? So I've highlighted the important bits, i.e. the mutant bits. Here we have a mutant promoter. Here we have a super repressor. And here we have a constitutive operator. They're in green because they're excited to be here. And here we have a dead Y structural gene. The important parts are that we've got a high concentration of lactose and glucose, and we're specifically talking about the Z structural gene. So, what are we going to observe? Well, The first important part is this. That first operon's P is mutated. It does not work, which means that it cannot interact with RNA polymerase, and none of this is going to be active. The whole first operon is going to be irrelevant, because no matter how excited this repressor is, no matter how healthy this operon is, RNA polymerase just can't interface. Not going to happen. We can ignore this one. Let's look at the second one. It's got a good promoter. Its operator can't be repressed. So even though it has a super repressor, that super repressor still can't bind to the operator. So we're going to have transcription. In fact, we're going to have constitutive transcription because it is not suppressible. This operator is going to continue to recruit things and will be happy to be here. However, the glucose present will prevent CAP-CAMP from enhancing transcription. So this glucose here will prevent the extra enthusiastic recruitment of RNA polymerase that CAP-CAMP usually does. So Z will be transcribed, though not Y, but we don't care about Y because we weren't asked about Y, we only care about Z. Z will be transcribed, but it will be transcribed at low levels because we have glucose available. Even though the superrepressor is there, and theoretically it should be repressing whether or not we have lactose, it can't interact with our constitutively active operator. However, there's glucose present, which will prevent CAP-CAMP from increasing RNA polymerase's recruitment. So we're going to have a very small amount of expression. 19, and I broke this down for A and B. Is there a C? Yeah, so I'm going to do A first and then do B and C together. So for A, question 19, briefly but accurately describe 4 different mutations that could decrease expression of the arabinose operon's structural genes. We're trying to find mutations that could mess up, decrease expression of arabinose. There are a bunch of different ways to mess up arabinose. Here's a few. You could mutate RSC, the gene that codes for the RSC protein, such that it cannot bind arabinose, which, if you remember, causes this inhibition. So RSC interacts with arabinose, causing inhibition, or it doesn't interact with arabinose, and when it isn't interacting with arabinose, it promotes transcription. So you can mutate RSC. So remember, RSC has those 2 different settings. It can be an inhibitor or it can be an activator. To decrease expression, you could either prevent it from becoming an activator by preventing the binding of arabinose. You could prevent it from activating by preventing it from interacting with RNA polymerase, or you could prevent it from activating by preventing it from binding to RI here. The other thing you could do is mutate RO so that it binds RSC even when arabinose is bound to RSC. So you could make RO kind of not know whether or not arabinose is there, and that would force RSC to be inhibitory all the time. You could also mutate RI so that it can't bind RSC to recruit RNA polymerase. Another thing you could do is mutate the promoter so that it doesn't work anymore. That would be simple. You could also mutate the structural genes so that they have early nonsense mutations or don't function properly. There are plenty of other things to do. Make sure you know the anatomy of this gene. Woo! Next. B and C. For B, the question is name 2 specific types of mutations that could turn a wild-type arabinose structural gene into a null mutant gene. Once we've talked about that, we're going to go more in depth. For one of those mutations, you're going to explain how it causes that change from a wild type into a null. So, just focusing on B, we're asking how does it turn a wild-type arabinose structural gene into a null mutant? In order to do that, you need to understand what null mutant is. And that's just something that doesn't work, does not make a functional protein. Almost any non-silent mutation would do. There's missense and nonsense and insertions and deletions and frameshifts. So as long as you can justify it in C, then you can say it in B. So for one of the things that you said in B, you have to say how. And I'm going to use missense as an example because it's one of the more complicated options up there. Here's our example, that missense mutation can change an amino acid found in the active site of the enzyme so that the enzyme cannot bind to its substrate. Therefore, no functioning enzyme is produced, and this is a null mutant. In order to get full marks for a clearly explained question, you have to say, it can do this specific thing, which results in this specific defect, which makes it a null mutant. You have to follow it all the way to its logical conclusion. Logical conclusion. All right, almost done. Home stretch. Question 20. The genes found in our human nuclear DNA never use the attenuation mechanism like the one observed in the tryptophan operon. To regulate gene expression, explain why in terms of the mechanisms of attenuation. Here are the important parts. I've thrown in an attenuation image for reference. Again, this needs to be in your head, this attenuation image. You should be able to draw this and make it make sense to yourself. So, we're specifically talking about our genes, i.e. eukaryotic genes, and our eukaryotic genes don't use attenuation. Why? This is the answer. It's because attenuation requires transcription and translation to be in the same place, because this termination loop is dependent on the location of the ribosome in reference to RNA polymerase. And these things are not in the same area in eukaryotes. Attenuation requires translation to begin before transcription is complete, which doesn't occur in eukaryotic cells because transcription and translation occur at separate times and locations. Question 21. Give the genetic symbols for two mutations involving the lac operon that would result in constitutive expression of the lac structural genes. Clearly describe how each mutation results in that constitutive expression. Okay, here, have a lac operon. You should be able to produce this from memory, this image. I'm going to explain two mutations. First is you can do an I-, a non-functional repressor. So this mutation would result in no functional repressor protein. So it might produce a repressor, but it would be unable to bind the operator sequence. Without the repressor protein to repress gene expression when lactose is absent, the structural genes would be expressed constitutively. This is the other one, OC. This is a constitutive operator. That's what the C stands for, constitutive. This mutation results in an operator sequence that cannot be bound by the repressor. So it's kind of the partner to the I- is the OC. That's the counter. It results in the exact same functional outcome. Since the repressor can never bind the operator, whether or not lactose is present, gene expression is never repressed, and the structural genes are constitutively expressed all the time. Okay, here we go. PEQ 13. I'm going to tear this thing apart. This is the whole question. Are you ready? You're studying a population of parakeets living on Island A. In an earlier experiment, you crossed pure-breeding yellow-beaked birds with pure-breeding white-beaked birds, and all of the offspring had pale yellow beaks. Recently, you surveyed all 1,000 of Island A's parakeets and observed the following beak colors. These. Is this population at Hardy-Weinberg equilibrium? Show your calculations and use the provided chi-squared table. My goodness, that's a lot. Let's boil it down. Starting from the top, this is the information that we got out of it. You're crossing pure-breeding yellow-beaked birds with pure-breeding white-beaked birds, and the offspring has pale yellow beaks. That's this. That's what that says. Yellow beaks crossed with white beaks equals pale yellow beaks. We've seen this before. This is partial dominance. Okay, next part. You surveyed 1,000 of Island A's parakeets, and this is what you saw. Okay, that sentence means this, right? So if we smoosh this all together, we're taking this partial dominance outcome that we're aware of, we're applying it to this knowledge here, we get this. Now we know the genotypes of these 3 phenotypes. And we know the number of individuals out of 1,000 that has each genotype, because we know that it's partial dominant. So here's the question part. Is the population in Hardy-Weinberg equilibrium? Show your calculations and use the provided chi-square table. Okay, Hardy-Weinberg equilibrium chi-square test. We're going to be doing one of those things. 1 foot in front of the other. In order to figure out if it's at Hardy-Weinberg equilibrium, we have to figure out P and Q. This is how I did that. P is legit just the number of big Y alleles added together and divided by the total alleles. So there are 110 individuals that have 2 of these. So we're going to take 110 multiplied by 2. There are 485 that only has 1. So we're going to add 485. Then we're going to divide that by the total alleles. There are 1,000 individuals, each of which are diploid. So they all have 2 alleles. So we're going to divide them by the total alleles, and this is what we get. We only have 2 alleles. So 1 minus that is this. So that's our P and Q. So now for our expected, assuming Hardy-Weinberg. So for a population in HWE with this P and this Q, our expected number of birds with 2 big Ys, with this YY phenotype, equals P squared, which is the chance to get a YY phenotype, 2 big Ys as a phenotype, times the number of chances you get, which is 1,000. So we take P, we square it, and then we multiply it by our N, which is the number of chances these birds get to have this genotype. And it's going to be this. Next, we're going to do big Y, little y, the heterozygote genotype, the heterozygote genotype, which is 2PQ, times the number of chances it has, 1,000. So 2PQ times 1,000, it's this. We could do the same deal for the homozygous recessive, just take Q squared times total birds, or you can take the 2 things that we have already and subtract them out of 1,000 to get this down here. So those are our expected. We're putting them in a chi-squared table with our observed. This is what we saw. This is what we expected based on our calculations. You need to be able to do this. You will not get away from this. I do this all the time in my research. This is your life now. Memorize these equations. You need to be able to produce this table here. Here's our P chi-square table thingy. We need to know our DF, which is the number of groups minus 1. We have 3, yellow, pale yellow, and white. So our DF is going to be 2. And if we take this 2 and we follow it over, we're looking for 3.9, which falls between 1.386 and 4.605, which is here between 0.5 and 0.1. which is definitely bigger than 0.05. Therefore, it is not significantly different from what we expected. Therefore, we are in Harvey-Weinberg equilibrium. Ta-da! That's how you apply a chi-square test to Harvey-Weinberg equilibrium. I would like to talk to you a little bit about studying. You have so much material at your fingertips. There's a million PEQs. You have all of my tutorial handouts. You also have all of Emily's tutorial handouts on that same page, the TA page on Canvas. Go look at it. All of Emily's videos are there. All of Emily's handouts are there. So go and check that out. You also have textbook questions and Mastering Genetics. There's also YouTube. Crash Course is a decent source. Khan Academy goes into way more detail, and I highly recommend them. Several of the pictures that I've shown you today are from the Khan Academy web pages. And there are just so many lectures on the internet. So many people have done this for you, this thing that I'm doing right here. And everybody has a different way of approaching these things and also different technologies at their disposal. And the vocab words. I will send them all out with this video. I will also post it on the Canvas page, on the Canvas TA page. Just a single document with all of the vocab words from the whole semester. Right there. Copied and pasted. Use them. Make flashcards. Explain them to your friends and families. Use them in paragraphs, stories, sonnets, conversations, whatever works to hammer them in so that you understand not just what they mean, but how they apply to each other and to this material. And get creative. If it doesn't feel like rote memory is working, stop doing rote memory. Make drawings and label them. Make paper cutouts. Do word scrambles. Google Images also has a bunch of different versions of all of the diagrams that you've seen in this entire course. So if one diagram is really not making sense to you, go find another one. And storytelling. I cannot emphasize enough how valuable it is to be able to use these concepts and turn them into a narrative, into cohesive storylines. Explain these things to your friends and families. Answer their questions. Get it to a place where you can describe this to a senior in high school, to someone who's only taken Bio 102 or 101. And study groups. I know in this world of social distancing, study groups are kind of hard to imagine right now, but please feel free to Zoom and Skype and make chat rooms on Facebook. Reach out to your classmates. Reach out to your group mates. Those discussion groups that I have on Canvas, you can make a discussion group for each other. You can talk to each other there if that's what works for you. Also, I'm gonna Zoom. I'm gonna have a Zoom office hour this Wednesday. Dear God. For you guys, that's probably going to be tomorrow, because I'm getting this out really late. So Wednesday at 3 to 4, details pending. You can also schedule small office hours throughout the day on Wednesday, and let's face it, probably Thursday. Email me if you can't make the Zoom meeting 3 to 4. I will send you a link or something to make Zoom accessible to you. Figure that out. Project for tomorrow. Woo! Yeah, project for Tuesday. That's a priority. Yeah, so that's the thing. I hope this helped. It has been an absolute joy to work with you guys, and stay safe and continue learning and asking wonderful questions. I am available for any other higher level questions that you have, or if you have questions about research careers or how to apply to labs, feel free to email me. I'd love to be a resource for you going forward. So yeah, have a great summer, and I'll see you around.